So it's my pleasure to introduce Dr. Xinla Xu. Dr. Xu is a research scientist at the immuno-oncology branch of the NCI at the National Institute of Health. He has 15 years of research experience in understanding tumor biology and immunobiology of human cancer. He's contributed to the understanding of mRNA payloads in tumor exosomes, through which melanoma cells can reprogram the metabolism of human stroma, a normal stroma. Prior to joining the NIH, Dr. Xu was a research assistant professor of oncology in Roswell Park, where he served as co-chair of the Exosome Working Group from 2017 to 2020. So without further ado, I will hand over to Dr. Xu. Let me share the screen.
So this is the overview. I would like to first talk about what are exosomes, and then on isolation of exosomes and do a comparison of isolation methods, its advantages and disadvantages, and then focus on reducing exosome-associated cytokines using a specific method. I'll also ask the question whether exosomes remain immunosuppressive even if all or most of the non-exosome cytokines are significantly reduced, and where do we go from here with this knowledge. After that, we will have a Q&A to hear your questions regarding exosomes and exosome isolations. So what are exosomes? I just want to clarify now that there is a lot of focus on getting the nomenclature correct on exosomes these days. So I would like to describe exosomes as a subset of extracellular vesicles. And here, the characteristics of exosomes are what I define exosomes is same as what the International Society of Extracellular Vesicles defined, which is that it has the presence or enrichment of tetraspanins such as CD9, CD63, or CD81. And then due to the nature of exosomes being created within multivascular bodies that are itself a specific size, the isolation method should be sized to less than 100 nanometers. It should also contain proteins that play a vital role in the endosomal sorting complexes required for transport machinery, such as DSG101. It is also known to carry protein and RNA cargo, and that is detectable only upon lysis of these vesicles. To give some perspective about size, here is an image from a scanning electron microscope. As you can see, these nodules that are appearing over here, they are about the size of multivascular bodies. Cells with high exosome counts appear to have these nodules. That seems to be consistent with transmission electron micrograph of exosome releasing cells. Except if you look at this, it looks like it's inside the cells. Maybe it is also found outside the cell. I've asked several cell biologists specializing in organelles, and I don't seem to have a clear answer what these structures are. If you know what they are, please let me know or shoot me an email. Anyway, back to exosomes. I'd like to explain further about exosomes and the tumor microenvironment. Because the tumor microenvironment is composed of many different types of cells, such as endothelial cells, immune cells, fibroblasts, and extracellular matrix. And there are vesicles released by these cells that surround the microenvironment, almost like a moat you see here. And these exosomes are mostly produced by tumor cells. These vesicles actually affect all cells in the tumor microenvironment. They are known to cause immunosuppression in all immune cell types, and you can read about this in the detailed review by Dr. Teresa Wanside. She publishes new and insightful reviews regularly, so I would recommend that you read her review to get a better understanding on exosomes, which she describes very elegantly. So, tumor exosomes, appear to be able to educate immune cells and stromal cells to promote cancer progression and metastasis. Our lab has also demonstrated that human melanoma exosomes can actually reprogram the healthy normal stromal cells. And one thing we need to know is that exosome payload itself isn't detectable using conventional means. And this will require separate isolation. So, for example, lysis. So, whether the exosome contains a cytokine payload, this is something that I am not going to cover. I am going to just focus on cytokines that are outside exosomes, so they are not related to exosomes. So we want to be sure that we get all the isolates as pure as possible, so that we can work with certainty that if the payload is studied, it is about the payload and not about an external co-elution. So technically, that's the bar we should set, even though the technology is not there yet to reach such high purity at this point. So if you look at this chart from our Frontiers Immunology review paper that we published recently, you can look at purity and you'll see that there are several methods that will give you high purity. So I would like to focus on two and discuss them in greater detail. So the rapid exosome isolation using ultrafiltration and size exclusion chromatography or RIAS is a method that we propose and hope more people use because it is able to provide high purity, and we do so and demonstrate this over several manuscripts. The other one is affinity capture method. This also gives very high purity. It is highly specific. So there are more research using this method to get so-called important exosomes. One concern of this is that we're still at a very early stage of exosome research, in the sense that we need to understand the payload better. So if you're already isolating a specific subset of exosomes, and we do not know the entire payload of all exosomes, and unless you affected in that other subset, will not influence the conclusion. The conclusion that you may drive may be affected and may be very different from what is happening within the tumor microenvironment. So looking at whole exosome isolate, I think it is a good place to start before you go into immunoaffinity method, if your hypothesis requires them. So I will explain the RIAS method in comparison with something that has been used consistently over the years, which is ultracentrifugation. And this method constitutes about 90% of how exosomes has been isolated over the past years, and this is because it has a lot of advantages over all the other methods. But there are some limitations here as well, which we will cover. And I would like to also share that on what RIAS method can improve upon the advantages that are demonstrated by ultracentrifugation by combining ultrafiltration and size exclusion chromatography. So so I tried to explain why I switched from isolating exosome using ultracentrifugation to RIAS. So the reason is pretty straightforward. It's a lot about time and effort savings, and without compromising purity and yield. I'm explaining this based on studies that involve the use of cell culture to get exosomes of sufficient quantities to work with them downstream. You can take the supernatant of the cell cultures, and then you do a low speed spin around 300 G, and then you collect supernatant, And then you do a higher speed centrifugation to get rid of the cell debris. And then you take a separator and filter for 0.22 micron series filter to get rid of any other debris that is above 200 nanometer in size. And then you load the tubes into ultracentrifuge machine, and you can go ahead and spin at 200,000 G for about 90 minutes. And then you can get an extracellular that's called pellet, which you can resuspend and quantify using NTA. So the total time it takes is actually about 140 minutes, or about 2+ hours to isolate and purify and quantify exosomes. So yeah, you can take the exosome out, and you can see that they have a fairly consistent diameter. You can examine these using tracking particle analysis, NTA, or dynamic light scattering, which is believed to be more accurate than NTA, but based on what I see, both are consistent in terms of their results. You can do scanning electron microscopy or transmission electron microscope. And you can see that the diameter varies a lot. For instance, you see this 130 nanometer particle, definitely not from a multivascular body. When you get these exosomes, you could actually quantify them using CD63 ELISA plates. For example, an exo ELISA plate. And these are customized for exosome identification and quantification. You can also use Western blot to see the CD63 that is enriched in these ultracentrifuge exosomes. One thing we have to note is that not all cancer cells produce a lot of exosomes. Because if you load the same amount of proteins, you see that CD63 can be fairly dim. Okay, so I will explain the Reyes method. So this method is optimized for benchtop centrifuge speeds. And the idea is that when there are cytokines and exosomes together in the supernatant, we try to get rid of them. The first few steps are similar. You do a few brief spins to remove cells and cell debris, followed by a syringe filter, 0.22 micron, and then you get a clarified supernatant that you can load onto ultrafiltration column of 100 kilodalton cutoff. It has been chosen for 100 kilodalton so that we can get rid of most of the cytokines and soluble factors that are under 100 kilodalton. And that would help to focus the concentrate more on exosomes and less on cytokines and other soluble factors. And the remaining job is done by the cell GS spin columns. And you will get a high purity exosome in the end. So you can read more about it in our methods in microbiology chapter. So the isolation of exosomes using size-exclusion chromatography, which is in the heart of Reyes method, you notice that that the morphology and the size is fairly consistent, and the human adult dermal fibroblasts, which represents normal exosome, they're a bit more sticky, so they get a bit higher. But yeah, overall, most exosomes are in the 50-60 nanometer range. Also note that normal cells don't produce as many exosomes as cancer cells, but this is our cancer cells that are known to produce a lot of exosomes. So to demonstrate further the isolation of exosome using size-exclusion chromatography, it is quite ideal to get the whole exosome instead of a subset. Is when you take a 100 kilodalton cutoff ultrafiltration, you actually get and CD63 band. And when you look at the flow through of the ultrafiltration, there is no band there. So you know that the ultrafiltration itself doesn't remove at least observable amount of exosomes, which is good. And this is also the case for 3 kilodalton size cutoff ultrafiltration columns. And that is also for flow through, it captures it pretty nicely. But we want to avoid this because you'll be getting all soluble vectors, such as cytokines, isolated together. Even if you put it through an SEC, you're going to get contaminants. And this represents normal, healthy exosomes, and this is the flow through as well. And this one would show that if you just put in ultrafiltration without doing a cytokine column, you'll notice that there is CD63 band over here. But look at the amount of albumin or other protein that actually comes in together when we load the Western blots. So bear that in mind that SEC is something that should be in your workflow. This lane is the cell lysate showing beta tubulin, and we showed the same Western for other cancer cell line derived and healthy exosomes for CD63, and there is a bit of beta tubulin contamination, but I think it's reasonable. Yeah, we can't avoid it completely. So if we look at it by comparing Reyes method with ultracentrifugation, you'll see that when you do a multiplex ELISA of all the key T-cell immunomodulatory cytokines such as GM-CSF, interferon, gamma, IL-10, etc., you will notice that a significant amount of cytokines are actually concentrated during the exosome isolation process. And R stands for Reyes, U stands for ultracentrifugation, and you can see that there is a very high concentration of cytokines present in ultracentrifugation isolates. And this is true for roughly 600E melanoma cell lines, and also for BRAF-type melanoma cell lines. And Reyes method does not completely eliminate any like cytokines that is present. It does leave some amount of cytokines, but most of them are under 100 picogram. I have no way to demonstrate if this is exosome-associated cytokines, or is this still part of something that's not related to exosome and is a coelute, but you can clearly see that it definitely brings a better purity into the picture. And with regards to where did all the cytokines go, we can examine the flow through. So these are concentric circles of tenfold difference. So they are like log 10 concentric circles. So you can see that the concentrate used for loading into SEC, which is the Reyes method, you get it within the first 2 circles. But the flow through is the one that you'll see that most of the cytokines are there. So we know the ultrafiltration columns does most of the job for removing cytokines. So after removing all these cytokines that are usually co-isolated with exosomes if you use just ultracentrifugation alone, are exosomes still immunosuppressive? And to answer this question, we can use a technique called surface plasmon resonance, or SPR, to detect exosomal PD-L1. So what they will do is, if we have more PD-L1 positive exosomes, we know that it will be immunomodulatory and possibly immunosuppressive. And we checked reflectivity of the antibody itself, we chose two most popular clones, MIH1 and 28H, and we found that it correlates, the normalized reflectivity correlates pretty well with PD-L1 concentration. So we using this method, we'll be able to determine how much or how many PD-L1 positive exosomes are there, and you can notice from here that Rios definitely has more PD-L1 positive exosomes than ultracentrifugation, and this is true for BRAF V600E melanoma cell line exosomes or BRAF multi-melanoma cell line exosomes. And this PD-L1 exosome, you can also detect it using NTA. So you can see that we can use PD-L1 antibody conjugated with Q-dots, which was chosen because NTA is a very powerful laser that they can bleach everything. Q-dots can extend the bleaching process. So when you use the NTA lasers, we can see that there is a PD-L1 positive exosome that we can detect. It is not a very large amount, but the signal is definitely there. You can also use image string, which when you load just the PD-L1 antibody only, you notice there are some contaminants. And when you add the human melanoma exosomes together with the antibody, you'll see this new area lighting up. But when you add in Tritonex, that vanishes as well. So this demonstrates that there is PD-L1 positive exosomes that we are able to capture and show using image stream, which is a flow cytometry machine. So with the advancement of chimeric antigen receptor or CAR T cells, I think the best functional immune assay for exosomes in relation to immunomodulation there is one that can answer the following question: does exosomes cause immunosuppression of antigen-specific CD8 T cells? So we had the opportunity to test the immunosuppressive effects of melanoma-derived exosomes on patient-derived NY-ESO-1 tetramer-positive CD8 T cells, which we will call NYT cells. So NY-ESO is a tumor antigen expressed in melanoma. And to set the stage for the investigation, we confirmed the identity of antigen-specific T cells by checking the binding of NY-ESO-specific T cell, NY-ESO-specific tetramer, and we got more than 90% tetramer-positive NYT cells. So these were challenged in vitro by exposure to melanoma cell line that is HLA-E2 positive and also express NY-ESO-1. We then examined the immune function by measuring the intracellular production of interferon gamma and interferon alpha. So in the absence of melanoma exosomes, co-incubation of NYT cells and NY-ESO positive melanoma cells between 4 hours caused most of the tetramer positive NYT cells to produce interferon gamma and a reasonable number of cells to produce TNF alpha. However, the percentage of interferon gamma producing NYT cells decreases significantly for BRAF wild-type melanoma cell line when cultured in the presence of BRAF V600E melanoma cell line 88 male or the BRAF multi-melanoma cell line 2183. So therefore, the percentage of interferon gamma and interferon alpha producing NYT cells was reduced by more than 50% when cultured in the presence of melanoma exosomes, regardless of the BRAF mutational status of the melanoma cells of origin. We also checked if the observed immunosuppression could be attributed to the loss of T cell viability due to exosomes using an XM5 PI analysis, but no noticeable change in the apoptotic fraction or loss in viability of T cells was observed. So these exosomes are clearly immunomodulatory, even without the presence of non-exosome-associated cytokines. So let's zoom out of the picture and see where we are in terms of cytokines and in relation to exosomes. Currently, I have only investigated what happens if you reduce cytokines that is co-isolated or concentrated with exosomes, but I did not examine cytokines within exosomes. I do not contest the studies of my colleagues that when you lyse exosomes, the cytokines are released and this increase is measurable. I'm only suggesting that it may be possible for cytokines to be co-eluted into exosome isolation. So this possibility should be factored into experimental planning, both upstream and downstream. So I'm here to suggest that the tumor microenvironment may have another layer of complexity that we may have not been examining in detail, which is the tumor exosome microenvironment. This sort of paints a more complex picture about the tumor microenvironment. So you can imagine there is an evolution perspective that cell-to-cell communication is important in tumor progression and metastasis, which we partly overlook its importance because the isolation of specific vesicles have been technically challenging. There are limitations to all exosome isolation methods, as I have mentioned, but we know that the tumor exosome microenvironment is filled with cancer exosomes, as you can see, and we have emphasized it using crayons. It is also able to gain access into the rest of the body through blood vessels and vessels. These also, you can see that there are immune cells within the tumor microenvironment, and they are getting a massive dose of tumor exosomes, and they themselves produce exosomes as well. So we have some knowledge about what kind of exosomes these reprogrammed immune cells produce, and it is supporting tumor growth and progression. So the environment is complex, and to study cell-to-cell communication in this microenvironment, we need to examine exosomes by themselves. So we have to be aware that cytokines and soluble factors are also being produced, and some of them are contained within exosomes. We need to also know where are they coming from, which cells are releasing them, to get a proper understanding of what these exosomes do, or how are they involved in tumor progression. So where do we go from here? So I'll suggest to exercise caution when isolating exosomes for downstream experiments, especially when you're looking into immune cells or immunotherapy-related experiments. You should probably consider providing a baseline cytokine profile. This is a transition recommendation right now. Maybe later, when there are more standardized ways to provide evidence regarding cytokine association with exosomes. I think some groundwork should be invested so that exosome studies can become clinically relevant. So I would like to finish my talk and thank Dr. Mark Armstrong, my wonderful mentor. He has been amazing in guiding my cancer research. He starts every lab meeting with "Did you cure cancer?" And I'll never forget that simple statement. It provides clarity of purpose in everything that I do in research. And I was able to do all this wonderful work thanks to him and also my wonderful collaborators, such as Dr. Hans Lindemann and his flow cytometry core in National Park, Dr. Teresa Whiteside, Dr. Richard Banquet, Dr. Yunu, and the electron microscopy unit from the University of Buffalo. Although the lab is now no longer at Roswell Park, I continue to look for opportunity to continue with this work. This research was supported by National Cancer Institute and Roswell Park Comprehensive Cancer Center. Thank you, and I would like to open the seminar to any questions that you may have.
So for the first question, Shin, do you know what the vesicles on top of the cells are? You showed an SEM image.
Yeah, so I was saying that I I think they are multivascular body based on the size, but I have asked several cell biologists, they do not know what these structures are. So my answer is I do not know, but these are highly exosome producing cell. I won't be surprised if they have special organelles to produce exosomes as what it looks like budding vesicles are coming out. That this is completely open to debate. I could, if anybody's interested in this image, I could send it over uh, to anyone that's interested to further investigate what these are. They have been fairly consistent, consistently found in cells that produce a lot of exosomes.
Okay, great. So again, if you'd like to get a copy of that image, just get in contact with us at Cell Guidance Systems, so tech@cellgs.com. Next question from Jim Shazad Hussein Prince. A couple of questions actually. The first part, is commercial benchtop exosome isolation kits as efficient as ultracentrifugation?
You're right. So there are a lot of commercial benchtop exosome isolation kits. Many of them are based on polyethylene glycol. Some they call it proprietary, but actually the most convenient ones are based on polyethylene glycol. The problem with that is that you do get exosome, but you do not get intact, like completely intact exosome, because due to the nature of this PEG-based activity, they actually sort of remove vesicles out of the water body by desiccation. So you actually get aggregation. You can see on transmission electron microscope of aggregated exosomes the size of 1 microns even. But you see, essentially, what do you want from the exosome? If you want like RNA out of it, they are, after all the vesicles. So if you are purifying RNA out of it or looking at proteins, you won't affect you at all. If you are using that to inject mice downstream, that's a no-no, I think. You don't want to inject coagulated stuff into mice. So I don't know what exactly is the exact isolation kit we are looking for, but yeah, the most common ones, are they as efficient as centrifugation? It's really hard to say what you are comparing with. I did give, our group has the review paper that that I got the table from, which I presented. You can take a look at that. If you have any questions, you can shoot me an email. I'll be happy to help.
So another question is, can you elaborate on how you can get a baseline cytokine level associated with exosomes? They may have missed it. So can you elaborate on that, how you get your baseline cytokine levels? Okay,
so the baseline cytokine level, what I did was by doing multiplex ELISA. You can read on my paper called on purity of exosome. It was published in a Journal of Extracellular Vesicles in 2020. So you can read further about it. What I did was to get multiplex ELISA that for T-cell panel. It can be quite costly, but bear in mind that if you are going to do exosome research and you have found a very suitable way that you want to work downstream, I think the cost is worth investing in to just make sure that your cytokine levels are acceptable.
Okay, another question that's coming through the chat from Raja Gopal, apologies for pronunciation. What was the initial volume you took for isolation of exosomes?
Right. So this initial volume is also in my paper, so you could read up on any one of them or the chapter. on our exosome isolation method. We generally take a full falcon tube amount. Yeah, so depending on how much we need, it can vary, but typically it can be between 15 to 50 mil even. We get a fairly consistent yield regardless of how much we start with, in the sense that they correlate pretty well with what is the starting amount. But yeah, they are between 15 to 15 milliliters. I generally don't go past Falcon tube size because things get a lot more complex.
So, question here: why do you think the REIUS method generates exosomes with more PD-L1 than ultracentrifugation? Your data seem to show that the REIUS method enriched for exosomes with PD-L1. Why do you think that is?
I've covered that in the REIUS, like the purity JV paper as well. It's a simple correlation due to yield. I show that it is we get a nearly 10-fold more yield compared to ultracentrifugation. So naturally there'll be more PD-L1 exosomes in there. It doesn't specifically like get more PD-L1 exosomes. It's just the general yield has gone up relative to the volume.
Okay. There's a question here from Yael Rosenberg Hassan. Have you measured and compared cytokine levels in plasma or serum baseline and from the exosomes isolated? Do you have any specific recommendations for plasma and serum?
So for plasma and serum, it is still very challenging because there are a lot of other issues involved. The main issue is with platelets. Our blood plasma contains platelets, and platelets are they can be very close to the size of exosomes. They're not exactly the same, so they are quite distinguishable. But there's been a question about should you use serum or plasma, but it really depends on what you want to work downstream. Based on my experience, either serum or plasma, I do not notice any difference. Although there's been a lot of theoretical argument that plasma is better because you don't coagulate things, and when things are coagulated, you actually lose exosomes in the process. But I think it's still a bit theoretical because the sheer quantity of exosomes, to give you a rough idea from Dr. Teresa's Weiss's paper, it's 10 to the power 12 particles per mil of blood. So I think it is really relative. Yeah, but at this point, if you're asking a more specific question like can size exclusion column like cellular GS exospin isolate exosomes using plasma or serum, I have no published data, but based on my experience, yes, they do a fine job.
Okay, another question is, can you comment on whether the cytokines that are eluting, do you think they're bound on the surface of exosomes, or are they just co-sedimenting during ultracentrifugation?
So you can actually read up on papers. I can't remember the specific paper that addressed it. What they did was to actually do co-labeling on the exosomes, and they found cytokines to be stuck on the membrane. So it is at this point, my research is meant to caution people that we need to keep progressing in terms of exosome isolation, because cytokines are so closely associated. If you go back in history, within soluble factors, there were extracellular vesicles, including exosomes. And then, it's only, after the 1990s, they become a very distinct proportion in the literature. So, yeah, it is that intricate alongside with other soluble factors like cytokines. It's hard to distinguish them. And my study is not meant to distinguish them. And if you are asking, can cytokine be completely isolated from the elimination of the other, like exosome and cytokine, can they be eliminated or become mutually exclusive? My answer is no, because I don't have any data to demonstrate that, at least within using my method, any of the cytokine profiles go straight down to zero. There is always, like 5-10 picogram remaining. It's really hard to say whether, they are associated with the exosomes.
Okay, I think time for just one more question. So in your analysis of cytokine levels between Rios and ultracentrifugation, there were some of the cytokines that were particularly elevated, like IL-10 and IL-18. Why do you think those cytokines might be particularly elevated? Is there anything to explain that?
So I can give one explanation for the IL-8. There are other cytokines, but bear in mind that these are supernatants of melanoma cell lines. So they actually produce a lot of cytokines, and what we are looking at is actually a concentrate or co-elution of a concentrate that wasn't completely removed. In a sense, when you concentrate using ultrafiltration, but most of the cytokines do fall off ultrafiltration, and some still remain. To answer your question, so there are two cell lines that I chose, which has a very important implication in melanoma, which is whether their BRAF mutation profile is V600E mutation or wild type. And there has been literature demonstrating that BRAF V600E mutant melanoma produces a lot of IL-8, and I think that is consistent with the literature. So yeah, the cytokine profile you see here is fairly consistent with what we understand about melanoma in the literature with regards to how is it associated with cytokines. At this point, it's very difficult to say whether these cytokines are stuck to exosomes. So yeah, more research needs to be done. But all I'll say is that if we can increase the yield by tenfold and still reduce the cytokine level so significantly, there is a good possibility that the exosome-associated cytokine may be smaller than we envision. But yeah, I can't comment more on that without any supporting evidence.
Okay, thanks very much. So I think we'll leave it there, Shinla. Maybe one last thing I should say is that the work that Shinla was describing here, the size exclusion columns that were used in that work were cell guidance systems exospin columns, and if you have a look at the exospin, the website, you can see those columns there. You will be getting, I think, a notification providing a link to a recording of this talk, and there'll be contact details for Shinla there as well. So if you have any further questions and you want to get into contact with Shinla, I'm sure he'd be happy to answer your questions directly via email, or please get in touch with us through the tech@cellgs.com email. So thank you very much for your participation and joining us, and thanks again, Shinla.